Digitalis purpurea is a species of flowering plant in the genus Digitalis, in the family Plantaginaceae, native and widespread throughout most of temperate Europe. It is also naturalized in parts of North America and some other temperate regions. The plants are well known as the original source of the heart medicine digoxin. Description: Digitalis purpurea is an herbaceous biennial or short-lived perennial plant. The leaves are spirally arranged, simple, 10 euro 35 cm long and 5 euro 12 cm broad, and are covered with grey-white pubescent and glandular hairs, imparting a woolly texture. The foliage forms a tight rosette at ground level in the first year. The flowering stem develops in the second year, typically 1 to 2 m tall, sometimes longer. The flowers are arranged in a showy, terminal, elongated cluster, and each flower is tubular and pendant. The flowers are typically purple, but some plants, especially those under cultivation, may be pink, rose, yellow, or white. The inside surface of the flower tube is heavily spotted. The flowering period is early summer, sometimes with additional flower stems developing later in the season. The plant is frequented by bees, which climb right inside the flower tube to gain the nectar within. The fruit is a capsule which splits open at maturity to release the numerous tiny seeds. Subspecies: the three subspecies of Digitalis purpurea are, D.P. Subsp. Purpurea euro most of Europe, D.P. Subsp. Haywardia euro Iberia, D.P. Subsp. Mariana euro Iberia. Hybrids: Digitalis ex fulva, Lindell. 1821. Toxicity: due to the presence of the cardiac glycoside digoxin, the leaves, flowers and seeds of this plant are all poisonous to humans and some animals and can be fatal if ingested. Extracted from the leaves, this same compound, whose clinical use was pioneered as Digitalis by William Withering, is used as a medication for heart failure. He recognized it reduced dropsy, increased urine flow and had a powerful effect on the heart. Unlike the purified pharmacological forms, extracts of this plant did not frequently cause intoxication because they induced nausea and vomiting within minutes of ingestion, preventing the patient from consuming more. The main toxins in Digitalis spp are the two chemically similar cardiac glycosides, digitoxin and digoxin. Like other cardiac glycosides, these toxins exert their effects by inhibiting the AT-base activity of a complex of transmembrane proteins that form the sodium-potassium AT-base pump. Inhibition of the Na+/K+- AT-base in turn causes a rise not only in intracellular Na+, but also in calcium, which in turn results in increased force of myocardial muscle contractions. In other words, at precisely the right dosage, Digitalis toxin can cause the heart to beat more strongly. However, digitoxin, digoxin and several other cardiac glycosides, such as ourbane, are known to have steep dose-response curves, that is, minute increases in the dosage of these drugs can make the difference between an ineffective dose and a fatal one. Symptoms of Digitalis poisoning include a low pulse rate, nausea, vomiting, and uncoordinated contractions of different parts of the heart, leading to cardiac arrest and finally death. Cultivation: the plant is popular as a garden subject, and numerous cultivars have been developed with a range of colors from white through pink to purple, such as Dalmatian purple. Cultivated forms often show flowers completely surrounding the central spike, in contrast to the wild form, where the flowers only appear on one side. D. purpurea is easily grown from seed or purchased as potted plants in the spring. The following selections have gained the Royal Horticultural Society's Award of Garden Merit: the Shirley, Excelsior Group, D. purpurea F. Alba, Digitalis purpurea is hardy to zones 4 to 9. Uses Digoxygenin is a steroid found exclusively in the flowers and leaves of the plants Digitalis purpurea and Digitalis lanata. It is used as a molecular probe to detect DNA or RNA. It can easily be attached to nucleotides by chemical modifications. Dig molecules are often linked to uridine nucleotides. Dig-labeled uridine can then be incorporated into RNA probes via in vitro transcription. Once hybridization occurs in situ, RNA probes with the incorporated Ig can be detected with anti-Dig antibodies conjugated to alkaline phosphatase. To reveal the hybridized transcripts, alkaline phosphatase can be reacted with a chromogen to produce a colored precipitate. Genetics. The colors of the petals of the Digitalis purpurea are known to be determined by at least 3 genes that interact with each other. The M gene determines the production of a purple pigment, a type of anthocyanin. The M gene does not produce this pigment. The D gene is an enhancer of the M gene, and leads it to produce a big amount of the pigment. The D gene does not enhance the M gene, and only a small amount of pigment is produced. Lastly, the W gene makes the pigment be deposited only in some spots, while the W gene allows the pigment to be spread all over the flower. This combination leads to 4 phenotypes, M/, with / equals a white flower with purple spots. MM / equals an albino flower with yellow spots. M/, WW DD equals a light purple flower. M/, WW D/ equals a dark purple flower. In literature and music, this plant inspired a famous poem by the Italian poet Giovanni Pascoli titled Digitalis purpurea. It also inspired the Italian industrial metal band Digitalis purpurea. Gallery. References. Bibliography, Flora Europaea, Digitalis purpurea, Ecological Flora of the British Isles, Digitalis purpurea, Sky Flora, Digitalis purpurea, Purple foxglove USDA invasive plants of the U.S.